Welcome to Huwel Lifesciences, a leading Indian molecular diagnostics company focused on advancing accessible, reliable and affordable healthcare technologies. In this video, we demonstrate the workflow of the Quantiplus Lyo MDR-TB fast detection kit on open RT-PCR platforms. The Quantiplus Lyo MDR-TB fast detection kit is a real-time PCR assay designed for qualitative detection of Mycobacterium tuberculosis complex DNA and multi-drug resistant tuberculosis-associated mutations directly from sputum samples using an extraction-free workflow. The kit contains ready-to-use real-time PCR lyophilized amplification beads for the detection of MTB and MDR-TB, positive controls, molecular biology grade, purified water, quick lysis buffer with sterile swabs. Always verify the lyophilized bead labels before starting the assay. Additional materials required include Huwel Thermosonics device, micro pipettes with filtered tips, and a compatible real-time PCR instrument. The sputum sample collected in a sterile container must be properly labeled. Refer to the sample collection, transport, and storage guidelines for detailed instructions. Use only the sterile swab provided with the kit for sample collection. Do not use any other swab, as it may impact assay performance and test results. Take the sterile swab and carefully rotate by dipping into the sputum sample collected in the container for 10 to 15 seconds. Insert the sampled swab into the vial containing quick lysis buffer and ensure that the absorbent tip of the swab is completely immersed in the buffer. Twist the swab head against the bottom and sides of the tube 10 to 12 times to release the sample into the buffer. Press the swab head against the side wall of the vial and break the swab stick at the designated breakpoint. Do not remove the swab from the vial during the heating process. After mixing, ensure the swab head remains inside the vial containing the buffer. Dispose of the broken swab handle safely in a biomedical waste container according to laboratory biosafety guidelines. Place the QLB vial containing the sampled swab into the Huwel Thermosonics and run the predefined program in the device. After incubation, remove the QLB vial from the Huwel Thermosonics device and allow it to cool. Briefly centrifuge the vial. Now the QLB vial contains DNA, suitable for testing. Take the PCR tube containing the lyophilized MDR-TB amplification bead. Add 20 microliters of the prepared lysate directly into the PCR tubes containing the lyophilized amplification beads for MTB and drug resistance detection. The bead will dissolve to form the complete PCR reaction mixture. Close the PCR tubes securely. Briefly centrifuge to remove air bubbles before loading into the thermal cycler. Include positive and negative controls in every run to ensure accurate results. Place the prepared PCR tubes into the real-time PCR instrument. Use the preset program provided in the instrument and select the sample type as unknown, positive control, or NTC. Confirm sample positions on the instrument, verify the run parameters, and initiate the real-time PCR run by clicking the Start button. Use Huwel MDR-TB detector software for automated analysis and interpretation of real-time PCR data of MTB and MDR-TB detection. Amplification data generated from compatible real-time PCR instruments, including Biorad CFX96, Thermo Fisher Quant Studio 5, and Huwel T9 600 PCR systems can be imported into the software for automated analysis. Instrument-generated real-time PCR data are exported in Excel file format from the real-time PCR instrument. The exported Excel file is imported into the Huwel MDR-TB detector software for automated result interpretation. The Huwel MDR-TB detector software automatically analyzes real-time PCR amplification data and interprets MTB positivity, rifampicin resistance, isoniazid resistance, and MDR-TB status. The final interpreted report can then be reviewed, validated, archived, and exported for documentation purposes. The Quantiplus Lyo MDR-TB Fast Detection Kit simplifies molecular tuberculosis testing through extraction-free sample preparation and lyophilized PCR technology. Designed and manufactured in India by Huwel Lifesciences, the solution supports rapid, reliable and decentralized molecular diagnostics for tuberculosis and multi-drug resistant TB detection.